Welcome back. This is our fourth and final video for Chapter 16. Here we're going to look at eukaryotic post-transcriptional gene regulation, protein stability, and how altered gene expression can lead to cancer. So remember that in eukaryotes, after you transcribe your DNA and produce messenger RNA, it's actually not ready for translation yet. We actually have formed pre-messenger RNA transcripts that have to be processed before we can translate the messenger RNA into our protein. So this is our post-transcriptional modification, and this step, or these several steps, can be regulated as well. So remember, during that processing of pre-messenger RNA to produce messenger RNA, there were three things that occurred. We added a 5' GCAP, a 3' poly A tail, and then finally we had to splice out those introns, those intervening sequences between our important exons. So in splicing those introns, that's one of the first things that we can control in terms of gene expression. So here, I can see this picture from our book. I have 3 segments or 3 exons. These are the regions that will be important in my gene, my messenger RNA, and we want to get rid of the introns. During that process, we can regulate gene expression because we can make different proteins depending on which exons we decide to keep for our final messenger RNA product. In this picture, they show us 2 options. There could be more, but only 2 are shown. It looks like for the mRNA on the left, exon number 4 is missing, and that could generate a unique protein after translation. For the one on the right, we're missing exon number 2, and that could generate a different protein that will be translated. And there could be other combinations not shown here, maybe 1 and 2, or maybe one with 1, 2, 3, and 4, all 4 exons in there. This is known as alternative splicing. So our pre-messenger RNA can be alternatively spliced in different ways to create these different proteins, and this occurs inside the nucleus. But make a note that if you just look here at my 2 examples, the exons stay in order in terms of their sequence. You can't go out of order, so you can't reverse, for example, 1 and 2. You cannot have something like 2, 1, 3, etc. And that is just something that we've seen in all of the patterns for alternative splicing. How could have alternative splicing evolved? So what do we think? Where did it come from? We know that splicing requires the identification of introns. We have to make sure we don't mix up our exons and introns, and we properly cut out the entire intron for each of these. If there are errors in splicing, you could accidentally cut out an exon, which probably happened at some point in time. Usually this could be deleterious because this could be a segment of the mRNA that was really critical for the protein to function properly. But in some cases, it could have created a variant of the protein that allowed a new function or a modified function that was still adequate or even better or beneficial to survival. So if there was some kind of adaptive advantage to the organism to have this variation in these exons, then that would have been kept by the cell. And that could lead to an evolution to what we see today for alternative splicing. Besides having that splicing, remember that during pre-mRNA processing to messenger RNA, we also had that 5' GCAP, the methylated guanosine cap, and then we had our poly A tail on the 3' end. Both of these structures prevent the degradation, the breakdown of the mRNA by enzymes known as exonucleases. They kind of remind me of like Pac-Man, where they will just chop up your messenger RNA from the ends towards the middle. So luckily having that 5' GCAP and that 3' poly A tail prevents that from happening. So I'm going to erase my marks here. If you look closer at the mRNA molecule here, we also see regions that are labeled 5' and 3' UTR, and that stands for untranslated regions. These are areas in the mRNA that don't get translated into your polypeptide sequence. What these are important for are probably the stability or the longevity of our messenger RNA molecule. It turns out that there are proteins called RNA binding proteins, or RBPs, that bind to these regions, and they appear to influence the stability of my messenger RNA. They can either increase or decrease the length of time that we have the messenger RNA in the cytoplasm. They can also be involved in telling the mRNA where to go and how it will be translated, how long you get to keep the mRNA stable before it gets degraded by different types of enzymes. And in addition to those RNA binding proteins that bind to the untranslated regions in those 5' and 3' areas, we also have these elements known as microRNA molecules that recognize and bind to specific sequences of messenger RNA and can impact their stability and lifetime. So these tiny microRNA molecules will be complementary in sequence to the mRNA of interest, and miRNAs bind to or associate with a protein known as RISC, RNA-induced silencing complex. And this is a protein that includes our RNA in there. So the combination of microRNA and RISC, that complex, will bind to the messenger RNA and cause it to be degraded. So if you get rid of your mRNA at this point, then it can no longer be translated into protein. So here's another way to stop gene expression by blocking the translation of your messenger RNA because you have broken that mRNA down. So let's jump to the next step of gene expression, translation. There are also ways to control gene expression at the translational level. And we're going to go through these 6 steps in the next slide by looking at the figure. So during transcription, we saw that many proteins are involved in forming that initiation complex, like those transcription factors. Similarly, in translation, we also have many proteins that bind to and initiate translation. And we're going to form something known as the initiation complex for translation. So for translation to happen, we need a protein known as eukaryotic initiation factor 2. This EIF2 protein binds to the small ribosomal subunit. So remember, ribosomes are made of a large subunit and a small subunit. So when GTP, it's not really shown very clearly here, when GTP is bound to eukaryotic initiation factor 2, then collectively this complex is bound to the small ribosomal subunit. The next thing that's going to happen, which is not shown here, is that the transfer RNA that's connected to methionine, remember that start codon AUG that encodes the amino acid methionine, that tRNA is going to come in and it's going to bind to this whole complex, the EIF2 and the small ribosomal subunit. The next step is when GTP is going to be hydrolyzed and converted to GDP and inorganic phosphate. That releases energy, and we're going to see that the whole complex is formed. Eventually, this EIF2 will be released, and we'll have, it's not shown here, but the large ribosomal subunit will come and bind, and we get translation to occur by having the ribosome move forward on the messenger RNA and start generating that polypeptide chain, that chain of amino acids. Another way to regulate gene expression is at the protein level. If we don't want the protein anymore, we don't want to use the protein, we want it to go away, then we can mark our proteins for degradation. And we add chemical tags to proteins to affect their lifespan. Here we have a tag known as ubiquitin, and whenever we add ubiquitin to proteins, this allows them to start the process of getting degraded, getting broken down, so it's no longer going to be present. So this is really a mark for protein degradation, and it shortens the lifespan of that protein. It's now complete. What happens is that the protein will enter this complex known as a proteasome. It kind of looks like a trash can almost. And this will chop up your protein complex so that you can reuse those amino acids to build other proteins. So these proteins are chemically modified, as we can see, and that changes the lifespan or longevity of the proteins in the cell. These changes can alter epigenetic accessibility, transcription, mRNA stability, or translation. So this refers to the idea that we've seen throughout the chapter where these changes refer to chemical modifications, these chemical tags. Earlier we saw methylation, acetylation, and now we're looking at adding ubiquitin to proteins. Towards the end of chapter 16, you'll see a section on cancer, because cancer can also arise due to altered gene expression. Cancer is usually not considered a single disease, but many things that have changed within the cell to allow it to divide uncontrollably and no longer perform the normal cell functions. So usually this results in a combination of different mutations that have accumulated within the cell. The cell can no longer have a regular cell cycle, and again, the cells don't stop growing as they should. What we often see is that in cancer, we have overexpression of certain genes or expression of genes that are not normally expressed in a cell. And this can be due to mutations in a gene or different combinations of genes, or it can be due to what we've been focusing on in this chapter, epigenetic changes or some kind of impact on the regulation, the normal regulation at these many different stages that usually occurs in eukaryotic cells. One example is something we've seen before, tumor suppressor genes. These encode proteins that are going to function to inhibit the cell cycle from moving forward. So these usually prevent excess or inappropriate cell growth. It turns out that one of the most popular and well-studied tumor suppressor genes is P53. It turns out that in over 50% of all cancer types, there is a mutation in this gene. So it no longer stops the cell cycle, and these cells are dividing uncontrollably. P53 also functions as a transcription factor. And I should include that we usually think of transcription factors as promoting cell growth because we're promoting the expression of genes. But the way that P53 acts as a transcription factor is that it activates genes that are going to be controlling the cell cycle and really inhibiting the cell cycle. These genes will encode proteins that inhibit the cell cycle. And this is especially true if cells are exposed to some kind of damaging radiation or other chemicals that have damaged the DNA. So when P53 is mutated, it can no longer inhibit the cell cycle, and that's when it causes or can lead to inappropriate cell division and cancer. In a previous chapter, we also talked about proto-oncogenes. These are normal genes that encode proteins that promote moving forward in the cell cycle. So they promote cell division. But when they're mutated, remember we call them oncogenes. And these can lead to cancer because they promote the cell cycle and they will not stop the cells from dividing or moving forward in the cell cycle even when they're not supposed to. So oncogenes can alter transcriptional activity of genes that control cell growth, where these genes are always on, even though they're not supposed to be on and the cell is supposed to stop dividing. One example is MYC or MYC. This is a transcription factor that's activated in a specific type of cancer known as Burkitt's lymphoma. And this is a type of B cell cancer. B cells are a type of white blood cell in your immune system that you'll learn more about in a future biology course. We can also have mutations in the DNA that turn out to activate transcription factors and promote the cell cycle when they're not supposed to. One example is in a certain subset of breast cancers, there is a mutation that causes the overexpression of something known as the epidermal growth factor receptor. So these are receptors on the surface of the cell, and if you have too many of them, that tells the cell to grow more, or to continue to divide even when it's not supposed to. And how it happens is molecules that bind to the receptors activate protein kinases within the cell, and that leads to the overexpression of transcription factors that promote the cell cycle. So what's kind of neat is that nowadays our sequencing of genes is so great and accurate, that we can look at cancer cells and look at where mutations are arising and directly address those mutations. Drugs that prevent the activation of these epidermal growth factor receptors, for example, are useful in treating this specific type of cancer. So this picture comes from another book, but it really is a nice example of how we can use genomics and DNA sequencing to profile cancer these days. Breast cancer, for example, is a heterogeneous disease. There are many different types of breast cancer and tumors, and we can sequence a tumor and we can look at all of the different mutations that are happening in that tumor. So here we have, for breast cancer, there are really 4 major types of breast cancer that are more common. And we can look at the DNA sequence and figure out where the mutations are happening and how to target those specific mutations. And that takes us to the end of chapter 16. In the next chapter, chapter 17, we're going to be looking at biotechnology and genomics.